have diseases that affect, we have diseases that affect the fruit, like scab or anthracnose. We have some branch dieback that can cause fruit drop. We have several beetles that are causing branch dieback. We have Phytophthora root rot. We have Botrytis branch dieback. So there's a lot of pests and diseases that will affect the yield and the quality of the avocado production. However, none of this is as devastating as Laurel Wilt. These are satellite images just acquired with Google Earth, and you can see how many trees we have lost. This is the Homestead area, which is the main avocado production area in South Florida, and you can see all these many trees, complete rows lost due to Laurel Wilt. Now, again, we have lost over 200,000 trees due to this disease since it was introduced into the area, which was in 2012. And here are some photos of what you regularly can see if you drive around Homestead. So it's pretty sad to see many plots sometimes abandoned or sometimes just trying to rehabilitate it, but you can see all these diseased trees around the area. It has also affected backyard avocados. It is very common for citizens of South Florida to have avocado trees in the backyards. They usually have one mango and one avocado. That's kind of like the norm. And when you drive around, you can see how these trees are also dying. And not only that, but in my belief is that they are helping the pathogen and the beetle move from one growth to another. So those are kind of the bridges that have contributed to the expansion and the epidemics that happen here. Also, the germplasm collection are being affected by this disease. We have lost several cultivars now in Trek because of this disease. And again, these are some of the photos of plantings being affected by the disease. Very common to see trees left in the field, completely dead, or some of the trees that have been stamped and still leaving disease stumps in the field. Thousands of acres have been lost due to Laurel Wilt, but our industry is resilient and growers are replanting. So what's Laurel Wilt? Laurel Wilt is a disease complex. We call it complex because besides the host and the pathogen, there's a third organism involved, which is the vector. But the causal agent of the disease is a fungus in the Ophiostomatiales group, and it's called Harringtonia lauricola. That's the new name. The previous name and the synonym is Rafaela lauricola, and that's the name I will be using in this presentation. It's a vascular pathogen. It has two ways of growing. It's dimorphic. So you can see here in the bottom of the slides, you can see the hyphal growth, but it also has a yeast growth, which helps the fungus move through the vascular system. This is considered to be a very virulent fungus because a small initial amount of inoculum can trigger disease and symptoms in the host. So here you can see a graph of artificial inoculation experiment that only 39 CFU, colony-forming units, it's enough to cause the death of 4 out of 6 mature trees. So this is very important. Why? Because it tells you that it's only needed a very small amount of initial inoculum, and the fungus is going to reproduce extensively and profusely within the vascular system, reaching the threshold for disease induction. So this fungus is being vectored by the Redbay Ambrosia Beetle, Xyloborus glavatus, and this beetle is the main vector or the original vector. And it carries the fungus in pockets called mycangia. And in the case of this beetle, they are in the front of the beetle. And then the beetle is going to create these galleries, going to bore into the trunk of the host, and it's going to farm and it's going to inoculate the galleries with the fungus. and the larvae is only going to feed on this fungus. So this is very important when we talk about management, right? Because the beetle and the offsprings are not eating the wood. They're actually eating the fungus, which is eating, getting the nutrients out of the host. So this beetle is different than other ambrosia beetles in that it infests apparently healthy trees. It's not attracted to ethanol, which is important to know in case you are planning on doing some trapping. The female mates before leaving the galleries and is ready to start a new colony. If the female is not fertilized, it can lay male eggs and mate. But recently I heard from the entomologists that 99.9% of the beetles, female beetles, are already fertilized when they leave the gallery. So a single female can start an outbreak. And this original vector, or the main vector, carries a lot of spores. In average, 6,000 CFUs. So a single inoculation can cause a mature tree to die. And the good thing is that this species is not attracted to avocado. However, it will bore into avocado if there's no other hosts. Now, the bad news is that in Florida, there's more than 10 species of ambrosia beetles that are contaminated with the fungus. So once the disease arrived to our area, and there were so many hosts infected with the fungus, these other beetles that were visiting these plants as well, boring galleries, coexistent with the Redbay Ambrosia beetle, got contaminated. And at least 3 of them have been shown to efficiently transmit the fungus into a healthy host. You can look for more information on this publication. So this disease affects members of the Lauraceae family, and at least 30 species have been proven to succumb to the disease. So what happens? This is a very simple diagram that I made to show you how the disease happens, right? You have your xylem vessels with the water flowing from the bottom up. Then you have your beetle that is going to make a gallery in order to lay eggs and cultivate the fungus. As it's doing this fungus escapes those galleries and gets into the stream of the vascular system and the xylem, and then it just spreads throughout. Now, once the host realizes that it's being colonized by the fungus, the defense mechanism, which is the production of tylosis, comes into play. And finally, it really clogs out the xylem vessels, impeding the water to flow. And what we get is like the host chokes itself, so stops the water transport through the vascular system. Now, this is a very good mechanism overall, very common in hardwood trees, and it helps the tree prevent the systemic spread of fungal that can cause decay. So it's a very good mechanism. However, in American Lauraceae, like avocado, the host reaction to the invasion is too slow and too profuse. So it results in extensive vascular damage and consequently death because of the lack of water. So it dies because of drought. Now here you can see how, as the time passes, this is artificial inoculation of potted plants. And you can see as the days go by and as the fungus moves through the vascular system and multiplies inside the vascular system, the vessels will occlude more and more. The more purple they are, the more water it passes with the dye, the less you'll see that the water is not passing, so the dye is not getting into these vessels. Now, the same happens in the field. These are artificially inoculated trees in the field, and you can see that the first symptoms are the loss of turgor because of the water has been stopped from preventing from moving. Then you have desiccation, and you finally have completely blighted, completely dry. The leaves usually stay attached to the branches, but if you get a little bit of wind, then you will lose the leaves. The internal symptoms, and Monica is going to talk in detail about this, but just to let you know, they are pretty dramatic, depending on how many days it has passed since the inoculation or since the natural infestation. And you will see a lot of these death of the vascular system. Now, when you make a cross section, you will see a lot of these rings and staining, very typical of this disease. Now, whether you have little spots here and there, or if you have the complete ring of xylem death of the new xylem, will depend on how many days since the tree was inoculated, and that's related to how you see